my first live session for this course, Introduction to Dynamical Models in Biology. We are in the third week of this course. By now, we had two other live sessions. You must have attended them. These were conducted by one of our TA, who is a PMRF fellow. He is organizing every week. He is organizing these online session for problem solving. Today also at 7 o'clock we have that session. The whole idea of that problem solving session is that in every week we are providing you additional practice questions, and we expect that you will try to solve them. And Achyuta, the PMRF fellow who is helping us in this course, he every week is meeting at 7 o'clock on Wednesday, Tuesday, and he is explaining all those problems, right? So please attend that if you have missed last two. The recorded video of those sessions are available on the course portal. You can go back and check. Before I move further, let me tell you that if you cannot hear me or if you cannot see me, please raise your hand so that I can understand that you cannot hear me or you cannot see me. And I hope you can see my white screen. If you have any problem, please raise your hand so that I can understand. Now, so apart from that live session where Achyuta is discussing about the problems that we are giving, in this live session, what I will do, I will discuss the broad outline of the course, and I will, for each session week course, I will discuss the key point that you should keep in mind, right? And at the end, I will discuss about certain tips that you should keep in mind for preparation of the final examination. Now, another thing is that if you have any query in between, please raise your hand and ask the question. There is a no, you can stop me anytime and I'll be happy to take that question. So let me start with the first week's topics. So in the first week, we have certain key points that I have jotted down here, which you should focus on first week. So in the first week, we are warming up. We are discussing about what we essentially mean about mathematical models in biology and why we need to create mathematical models, right? Those you may understand may not come as a question in examination or quizzes, but there are certain important things that you should focus on. Remember that every mathematical model that we are using in this course are based on ordinary differential equations, right? So ordinary differential equations in a model represents the rate of a process. That's what is written here. So what you have to understand in these lectures, the first week, is that how we represent the rate of a process using an ordinary differential equation. And then at the same time, you have to also focus, which is a very common question in examinations and quizzes and interview, is that, see, when we can use ODE-based model and when we cannot use ODE-based model. Ordinary differential equation-based model are called deterministic model. And you use two key assumptions while using it. And you have to understand those two key assumptions. We have discussed those in length in the course, right? So these two assumptions that the system should be homogeneous, meaning will mix. At the same time, the system size, that means the number of molecules, number of cells, number of organisms that you are modeling must be very high. Their concentration should be very high. That you have to understand. Also, you have to understand different types of ODE types, right? Linear, non-linear, and first order ODE, second order ODE, these things. These are the common questions usually we ask in interviews, exams, quizzes. Now, one important thing that you learned in the first week is that you create model inspired by law of mass action. Now, you must have studied law of mass action in your biochemistry book or in school text also, in chemistry also. We have to remember that law of mass action is primarily for elementary reactions. basic reactions. Unfortunately, when we create mathematical model in biology, we do not go down at any elementary level of reactions, right? We club multiple processes together. For example, let me give you one example. Say you want to create a model for transcription, and you will learn how to do that. And from the third week course, third week lecture, we are starting to teach you how to create ODE for transcription, how to write a ODE for enzymatic reaction and all these things. So you must have noticed that in that case, you see, transcription involves hundreds of molecules, different enzymes, but we are not breaking down that process into those hundreds of steps. We are clubbing all of them together. So that system is no more an elementary reaction. But still, we get inspired from law of mass action and try to write models based on ODEs using the concept of law of mass action. So in the law of mass action, you should know how mathematically law of mass action is represented. And there are some critical things. For example, if the reaction has, suppose something A of A plus B of B gives rise to C of C plus D of D, then you have to remember how to understand how to write the rate of this forward reaction in terms of the concentration and the stoichiometric constant. So this part you should be very clear on. And I have seen over the years students many a time make mistakes in that. So please look into that lecture where we discuss law of mass action and try to write differential equation for rate of reactions using law of mass action. Subsequently, in first week, we moved to disease model, and we also introduced you to something called logistic function, which is widely used in population dynamics, and in fact, not just in population dynamics, in other modeling systems, other dynamical systems also. So you should understand that logistic function. If you remember that logistic function is of this form, x into 1 minus x by c. So in this case, you should understand why we are using this part, 1 minus x by c. You should understand it very clearly. You should understand what is the meaning of this c. C we call carrying capacity, right? So you should focus on that and try to understand what does that imply? What do you mean by that? And how the value of this c affects the behavior in terms of this function, which we call logistic function. Subsequently, in first week, we moved into numerical methods. We teach you two methods. One is called Euler's method, which is almost 300 years old method and still going strong, and people use that. But the most of the time, the solvers that are used in solving large nonlinear ordinary differential equation-based systems are based on the Runge-Kutta method. You may not have to go in details of Runge-Kutta method for this course. When you are using JSIM in the background is actually using some form of ODE solver based upon the basic Runge-Kutta method. These are the advanced method. You do not need to go in details of it. But you should understand the principle of Euler's method by heart. And you should be able to execute Euler's method by paper and pen. You can easily do that. I have showed that in the video. Please try to practice. Take simple ODE or take example from the example that we have given in the additional questions. Take those ODE and try to simulate or numerically solve them using the Euler's method by paper and pen. You just do the calculation yourself. When you do that yourself, all the nitty-gritty of the method will get clear. Apart from that, what you should focus upon in this week is that you should understand the limitations of numerical method. When it goes wrong, how the time step size affects our calculation. That you have to understand. And from that understanding, you should move to the lecture where I have explained adaptive time step. So you should understand why we need adaptive time step. At the same time, you should look into the algorithm that is given in that lecture and try to understand every line of that algorithm. Don't mug up. You don't need to remember. Try to understand why we are doing that in that fashion. If you have any confusion in that, drop a question in the discussion forum or write an email to me or in the live sessions with Otsita, you ask this question, we are all will be happy to explain to you. So these are the key points that you should keep in mind for the first week's course of this course. So I'll just hold this screen for some time so that you can just go through one by one, starting from ODEs, ODE-based modeling, the different types of ODE, up to the numerical solver like Euler's method and Lunar-Kutta method. So these are the key points that you should keep in mind and focus upon while preparing for examination and quizzes based upon the lectures of the first week. Now, before I move into second week, is there any query from anyone? Anybody can ask a question by raising hand. Okay. Yeah. Is there any question? Okay. If there is no question, let me move forward. Before I move into the second week's topic and key point that you should focus on second week, let me tell you one thing. I have noticed over the years that sometimes learners who does not do well in first quiz, they stop submitting subsequent quizzes. Please don't do that. Remember that eventually the best quizzes are taken for your final grading, right? So if you even if you have not done well, performed well in the first quiz, you should always move forward and try your best for the second quiz and move on, right? So try those quizzes before the deadline and submit the quizzes within time. And then before quiz, you solve those quizzes. This time we are providing you additional questions also with answers and hints we have provided. Try to solve them. That will help you also in the quiz and the exam. So let me now move to the lectures of second week, and I will focus on the key point that you have to learn. See, this course is on dynamical modeling, right? Now, models, these are mathematical models. One way of understanding this mathematical model is to analyze this by paper and pen. Analysis of ordinary differential equation based model by paper and pen can be done to a large extent. If you are good in calculus, linear algebra, that helps and that becomes very useful. You can do those analyses by yourself. But many a time what happens is that large system, particularly coupled nonlinear systems that we encounter mostly in biology, are difficult to be analyzed analytically or by paper and pen. You have to rely upon numerical solutions, numerical method and graphical method. Further, all learners in this course may not be equally comfortable with calculus and linear algebra. For them, the numerical methods are easy way out, and using a numerical solver, you can easily understand the behavior of a model. Unless you simulate, unless you play with the mathematical model, believe me, you cannot learn that mathematical model. To learn a mathematical model, you have to play with it. I may have created a model in the lecture using a particular type of function in the ODE. Change those functions. Change the parameter values, simulate them numerically, draw the graphs and see how graphs are changing, how those behaviors of the system is deviating from what I have shown in the course. Now, there are lots of numerical solver available. If you are comfortable with programming, you can use Python, you can use C. If you have access to MATLAB, MATLAB has ODE solvers, you can use them. But if you don't want to go into programming and all these things, JSIM that we introduced in this course is a very good tool. It is in between. It also requires some amount of coding, but that coding is not like coding in C or in Python or R. It is coding like the way you write equations in your paper and pen using a pen. At the same time, it is a GUI, right? You can click, run, you can see the data getting plotted, you can save the raw data as text file, and then you can plot yourself in some other plotting software like Excel or something. So in a way, learning JSON is easy. So one video I have given where I have taught how to use JSON. And then subsequently, we are using JSON and we'll keep on using JSON for numerically simulating different models. So I'll insist please learn how to use. At least you learn JSON. Apart from, if you have interest, you may learn some other tools, but at least learn how to use JSON. It is very handy tool, very easy to use. His learning curve is very smooth. You can easily learn. And the best part is, if you go to JSON web page, there are lots of tutorial, lots of models given there. They have a library of models. You can download those models, play with them and learn. So I insist, please look into JSON. And remember, as in quizzes, as well as exam, you will have question based upon JSON. Although you have not to simulate using JSON, because in final examination, we will not provide you access to JSON, but you should know how JSON code works, how JSON works. So that type of question you should expect in the final examination. So that means you have to learn JSON by heart. So I insist, please go and learn. Again, if you find any difficulty, please post questions on the forum or ask me by email. Now let me go to some other topics which are critical for the second week. If you are revising and going through the second week's lectures, the key idea that we introduce in second week is steady state and stability of steady state. Please try to understand the idea of steady state and their stability by heart. There should be no confusion in you. What do you mean by steady state and what is the stability? What is the stable steady state? What is the unstable steady state? What is semi-stable steady state? This should be absolutely clear to you, because throughout this course, throughout lots of other models that we'll build in this course, in this third week and the fourth week, we will repeatedly encounter the concept of stable, unstable steady states in one dimension as well as higher dimensional problem. Right? So the idea of steady state should be very clear to you. Steady states appear ubiquitously in biological system. You take cellular processes, molecular processes, you go up to ecology, social behavior, everywhere you will find steady states. And these steady states can be stable, unstable. So we have to understand steady state to understand the behavior of a biological system. Steady state is one of the key types of dynamical features of a dynamical system, particularly in biology. Apart from that, one is oscillatory behavior. And in a couple of lectures, in fourth week particularly, we'll introduce you to oscillatory behavior also. Now, following the concept of steady state comes the question of direction field, phase portrait, and null clients. Now, these three things come together, what we call graphical method. As you can easily understand, these are called graphical methods because you are drawing diagrams, you are using graphs, plots, right? Now, these are the methods which has been developed long back when you didn't have computer and you do not have a computer to solve numerically a ordinary differential equation. And imagine, suppose we are back in 1960s, I do not have a computer, I cannot numerically solve a large ODE system, complicated ODE system. And it is such a complicated thing that I cannot solve it by integration, by numerical, analytical methods or linear algebra based method also. So how should I comment? How should I understand the behavior of this system? And that's where mathematicians developed the techniques of graphical method, where direction field is used for one-dimensional system. That means you have only one dependent variable, whereas the phase portrait and null clients are used for dimensions greater than one. Two-dimensional system, two variable, x and y, both depending upon time, or x, y, z, something like that, right? Or more than three. So more than one dimension when you have, more than one dependent variable when you have in your model, we use phase portrait and null clients based analysis. You may now ask, why should I learn them now? Because now I have computers. So you may ask now that why should I use them, right? That because I have now JSON, I can simply plug the equation, write down the ODE, and then simulate it. Why do I have to use this graphical method? Why should I learn them? The interesting thing is that this graphical method helps us to visualize the behavior of a dynamical system. And many concepts like steady state, oscillation, stability of those steady state becomes very clear when we visualize. That is why even now in a research paper, where they will use numerical method extensively to simulate a ODE-based model, they will still use these phase portrait and null climb plots because two reasons. One, using this plot, we can easily communicate a concept to our reader. At the same time, when you make this graph, when you plot them, many dynamical feature, many behavior of that system becomes much clearer to us. Just numerical solution and solving x versus t, y versus t data does not communicate everything. That's why phase portrait and null climbs are still important and people use them extensively. Following this, we introduce you to different types of phase portrait, like sink, source, spiral. Then we have the center type. These are very easy. Look at the phase portrait diagram and immediately it will click to you why we call a phase portrait as sink, why we call some phase portrait as source, why we call something as spiral sink or spiral source. Looking at the arrows, you can easily understand why we have given those names. You don't need to remember them, mark them up. Just try to understand the behavior of the system on the phase portrait, follow the arrows, give rise to your trajectories, and try to understand why we have those particular type of name. And remember, in many quizzes, exams, and interviews, people ask questions based on this type of phase portraits, right? So this is a very important topic. Phase portrait and different types of phase portrait is very important in the second week's lectures of our course. So I'll stop here because these are the key point of second week lectures that you should keep in mind and focus upon when you are preparing for exams and quizzes. Is there any question? Please raise your hand. I will take one by one. Can you people hear me? Samruddhi, can you confirm whether you can hear me?
Yes, sir, we can hear you.
Thank you. Okay, so what I will do now, I will move to the third week, this week's lectures and the key point that you should focus upon. So here we are. In third week, we have introduced a concept called bifurcation, and is very critical in understanding many exotic behavior in biology. And you must have heard of systems biology. The idea of bifurcation is extensively used in different aspects of systems biology. Bifurcation is a concept related to dynamical system. And in biology, we use this idea, this principle of bifurcation to explain diverse phenomena. For example, you must have heard of EMT, epithelial to mesenchymal transition. That happens in embryogenesis, as well as that happens in cancer cells when they undergo metastasis. Epithelial cell, they remain attached to each other. And when they get signal, like a signal by TGF beta, they detach among themselves from themselves. They become circular. They get migratory potential and they start migrating because they are no more epithelial, they are mesenchymal cell. Now I can consider epithelial cell as a phenotypic state, phenotype or type of phenotype for cell, whereas mesenchymal as another phenotypic state. The question is, how come these epithelial cells become mesenchymal? Your answer will be obviously that the gene expression must have changed. It's true, gene expression must have changed. That means the transcriptional regulatory process must be there behind this whole EMT that is driving this process. So even now, if you look into the data, you will find all epithelial cells will not all of a sudden become mesenchymal. Only some of them will become mesenchymal. So you can now ask, what is the difference between these cells that some of them has converted into mesenchymal, but rest of them has stayed back in epithelium? Or you can ask the question that why we only see this phenomena when TGF beta dose crosses a threshold? Why it does not happen beforehand? To understand this behavior, we now use the idea of bifurcation. and it is now widely accepted and extensively used. Similarly, an age-old question you must have heard is that how come embryogenesis happened? All of us at once upon a time was a single cell. From this single cell, different types of cell types has originated. And this happens by differentiation, and we know stem cell, we know there is transcriptional control. Now the question is, if I have one stem cell and it is giving rise to two different cell types, why one particular cell becomes cell type A, why the other cell type becomes cell type B? Why both of them are not same? What decides this? Is there any generic principle behind it? When I say generic principle, I mean that I don't want to look into specific transcription factors. I want to know generalized principles, generalized laws and rules behind this type of processes where one cell type now give rise to two different cell types, and some cell go in this path, some cell go in the other path. Why does that happen? This used to be an age-old problem. Embryologist has worked on it for decades, and now we try to explain it in terms of bifurcation. And in fact, you can create beautiful OD-based model for transcription factor like OCT4, nano. If you remember, OCT4, nano, these are the two critical transcription factors which are involved in the stem cells, differentiation of stem cells. So people have created mathematical model using the ODs the way we are creating model in this course. And then they have used the concept of bifurcation, the idea of bifurcation in those models to explain why some cell become cell type A and why other cell became cell type B. So we have to understand the concept of bifurcation in terms of ODs, differential equation, as well as what is their implication in biology. So please take your time. There are 2 lectures on that, but please repeatedly go through it, look into nitty-gritty of it, pay attention to everything that I am saying there. And then there are problems given in practice, problem given based on bifurcation. Please try to solve them also. And just to remind you, today at 7 o'clock, there will be a live session on problem solving for the 3rd week. So first important thing, and is extremely important for this course in this 3rd week, is idea of bifurcation in terms of mathematics of differential equation as well as its implication in biology. Then comes the question of network motifs. This is actually interesting, and many of you may have heard of network. The one problem, many a time, even our biology student face difficulty with it, is that so you open any biochemistry book, any book on metabolism, any book of cell signaling, you will see large diagrams. A controls B, B controls C, from C there is a feedback, and a huge diagram of pathways. They can be metabolic pathway, they can be cell signaling pathway, and nowadays we are generating large high throughput data. From that, people are generating pathways for transcriptional control. We have databases where you can actually get this diagram that who controls whom. But these are large diagrams, right? Large networks. Each of the node of that network is a molecule, and that get controlled or affected by something else. Now the question is, how can I understand the dynamics of such a big network? Suppose you want to take the insulin signaling pathway. How many molecules might be there in the pathway? If you boil it down, you may reduce it to 3 or 4 key molecules, but that's not the right way to look at it. There are maybe around 20 or 30 different molecules involved in the whole pathway. Can I create an ordinary differential equation based model to simulate the behavior exhaustively for this type of large network? The answer is no, for many reasons. First of all, when you create ordinary differential equation based model, you have differential equation. In that, you have dependent variable, independent variable time, and then you have constant terms which we call parameters, like rate constants, right? How will you know so many rate constants, so many parameters for a large model involving suppose 20 ordinary differential equations? You will not have those idea about those parameters. The first. Second thing is that you always will get trapped into this question. Do I know the network completely? Am I missing something? Is my model is complete? And the third problem that we'll face is that when you have a large model, then small mistake in your your model can actually lead you to completely in a wrong directions. And also computation time increases. Now, how can I handle this? Here actually one very interesting idea evolved in systems biology very early is that can I break down my network in small pieces? That's what people do when they analyze electronic circuit. An electronic circuit for suppose my computer or my phone may look very complicated. But if you remember, all electronic circuits are made up of certain bricks, right? For example, a resistance, a capacitor, right? Then a transistor, then a diode or a triode, something like that. So I can break down a large network of electronic component, which we call a circuit, into its parts. And each of these parts has their characteristic behavior. For a diode, you know what will be its input-output behavior. For a resistance, you know what will be its input-output behavior. For a transformer, you know what will be its input-output behavior. So the question is, can I break down a large biochemical pathway, which can be a metabolic pathway, which can be a cell signaling pathway or a transcriptional network, into components which will have their characteristic behavior. From there, the idea of network motifs arises. Network motifs are subpart or sub-network of large biological network which appear repeatedly, that means reused repeatedly, and has some characteristic behavior. Let me give you one example. For example, suppose A controls B, B controls C, means A activates B, B activates C, and C in turn inhibits activity of A. This will be negative feedback. And using simple ordinary differential equation based model, as well as by experiment, we know now that negative feedback has certain characteristic properties. What are those? Negative feedback can give rise to monostable system. What do I mean by that? If you have paid attention to our lectures on stability of steady state, then monostable means the system will have one steady state and that is stable. So negative feedback give rise to monostable system, or it can also give rise to oscillation. So this is a characteristic property of negative. So now I can imagine if you give me a large transcription network, I can break down these large network into parts and identify where I have negative feedback. And then I can say, okay, this negative feedback is controlling the expression of this gene. That means that gene may have oscillation or it can have some sort of monostable behavior. So in this way, rather than looking into the large network, what we do, we try to identify network motifs and try to model their characteristic behavior and then try to explain the behavior of the whole system in terms of these network motifs. Now, network motifs are sub-network. But even then, if you look into this, I have multiple processes, one process here, one process here, one process here. So I have three reactions, 1, 2, and 3. So but these three reactions, I have to write some function and ODs. How should I write them? And some people have asked earlier in the forum that, okay, you are showing us some models, but how do you write equations, OD of differential equation? How do you get the functions for those differential equations for a particular biological process, like cellular process, like transcription, translation, antibody binding to antigen, a ligand binding to a receptor, or a catalytic reaction is happening. Some protein is getting phosphorylated, some protein is getting dephosphorylated. How should I write the ODs for that? In this week's lecture, we have started. So what I have done, I have broken down all the elementary processes in biology, cellular and molecular biology in 4 parts. One is ligand receptor interaction, antigen antibody interaction, same thing, right? So we will learn how to write functions for ligand receptor interaction. Then another elementary process in any molecular and cellular process in biology is production and degradation of molecules. So that we will learn how to write a ODE, how to choose a function for representing production and degradation of molecules. And then we move one step ahead. Remember, proteins are created by transcription followed by translation. So there are dedicated functions to capture the control and dynamics of transcription and translation. So we learned that also in one lecture. And finally, we learned about enzymatic reaction. Although you can always argue that transcription also involves enzymatic reaction because polymerase is an enzyme. But we do not capture the dynamics of transcription by and translation by considering them as a separate enzymatic reaction using the functions like Michaelis Menten or something like that. We have something called Hill functions, which as a whole capture the dynamic of transcription and translation. And whereas in enzymatic reaction, for example, suppose a ligand has bound with receptor, suppose insulin has bound with insulin receptor, that trigger a cascade of enzymatic reaction where unphosphorylated protein gets phosphorylated, and that phosphorylated protein again act as a kinase to phosphorylate another protein. So you have a cascade of enzymatic reactions. These are very common in cell signaling and other molecular processes in a cell. So in that case, we should know what type of function I should use to represent those enzymatic reactions. So in this one of this week's lecture, we discuss. So these are the key points for this week's lecture, which are already live. I hope some of those you must have seen. Please go through them. Check out the practice problem. Try to solve them. If you find difficulty in solving any of those problems, get back to me, write to the forum, or ask question to Achyuta on his live sessions. For example, today he will have a live session at 7 o'clock. So that is what I have discussed till now on the key point that you should highlight or focus on for the first 3 weeks of our course. Before I move into the last part about the exam and preparation for final exam, if anybody has any question, please ask. Yeah, please go ahead. Can you unmute yourself? Mukunda?
Yes, sir.
Yeah,
Yes, sir.
please go ahead. Yeah, I can hear you. Please go ahead.
Sir, can we consider the cycle of degeneration of corpus luteum, the maturation cycle, into the oscillation system?
Can you come up again? Can you say clearly what exactly you are asking?
Sir, can we consider the menstruation cycle or degeneration of corpus luteum oscillation system?
As what system?
Oscillation.
I really do not know much about the physiology of that process. I have just broad school level idea, but I can give you a direction. The actually hormonal cycle, including the sex hormone cycles, right, are oscillatory. We know that, right, and those has been modeled very nicely long back. You have not, don't look into literature of last 1 decade. You can go back 80s, right? People have created very good model to understand the dynamics of those hormonal cycles using ordinary differential equation plus something else which we have not discussed in this course because that's an advanced topic. It's called control theory, right? So control theory is widely used in electronic devices and electrical engineering. You must be aware of, but our body also has homeostasis, right? Homeostasis is equivalent to steady state, and we have controls from starting from our control system in our brain to our hormonal cycle to gland. All these things have a nice feedback control mechanism which controls these oscillatory behavior of sex hormones and other things. So there are literature I know which I have seen for oscillatory behaviors of these hormones and how they are regulated, particularly the question that you have asked, I never looked into the literature of that, but I believe that must be there also. Is that clear, your question?
Yes, sir.
Yeah. Actually, there are excellent model using control theory. As this is a basic course that's why we have not gone. And nowadays, most of the people focus on molecular processes, cellular processes. We have now high throughput data, single cell data. All these things are coming. So people want to create models at molecular level. But remember, 20 years back, 30 years back, actually mathematical models at organism level, physiology levels were very popular. And we have beautiful model to explain how diabetes happens, and then how hormonal cycle works, how our sleep cycle works. Those are excellent mathematical models which are very generalized, and we have learned lots of things from them. And many of them actually people teach in physiology courses without talking about the mathematics. Thus they gist the basic things and teach to the students. Anything else, anyone?
Yes, sir.
Mukund, you have your hand raised. If you are done, you can talk.
Sir, can we have any reference book suggestions that include the mathematical models of such systems in our human body?
See, there are many textbooks. The problem is, first problem is that none of them are those are textbooks, right? All these are considered classically as a reference book, which are for researcher, and you need some amount of mathematical background to actually jump into them, right? Because those books are not written primarily for biologists coming from experimental biology background. Those are written primarily for mathematicians, electrical engineers, or physicists coming to biology and creating mathematical model. One book which is almost like Gita in the dynamical models, two books I will say. One is called Murray's 2 volumes of book in most libraries in major universities. If you have mathematicians, mathematics department working on mathematical biology, they must have purchased for the library. That is called mathematical models for biology, something like that, which is very well known. And then there is one book which is almost like Gita. If somebody want to create mathematical model, dynamical model, is Stroger's dynamical model, book on chaos and dynamical systems. So these are the books which everybody used to learn these things. But the problem is these are not accessible for newcomers, people who are entering into the thing, right? So what I will suggest you, if you are interested and if you are to some extent confident, I do not know your academic background, if you are well aware about calculus and linear algebra, these two are essential if you want to jump into even modeling these systems like hormonal systems or control using control theory to understand them. You need to have these basics in calculus and linear algebra at least. So then you can actually access those books. And if you are not accessing this book, what I can suggest you can look into my YouTube channel. There I have a series of lectures which are a bit advanced than what we are teaching here in this course, where all the mathematical details are given, and that will be a bit advanced. So if you are coming from a background where you have the mathematics background, I will suggest you to look into those. Fine.
Yes, sir. I'm a MSc Mathematics student from
Okay,
Maharaja
then you should.
Raja.
Then if you are an MSc Mathematics, then you have lots of books here. Let me show you one book which you should actually see. If you want to create dynamical model, this book on nonlinear dynamics and chaos by Stroger's is actually given. It has nothing to do with biology. You should always get back to it. It should be on your desk every time if you are working on that, because it has almost everything in it, right? And the best part is I will tell you, Stroger's still teaches using this textbook in his class. So, and those are recorded and presented on YouTube. So please search them and go through them, right? And then you access those books by Murray. And there are hundreds of other textbooks on mathematical biology. Mathematical biology is very rich in ecology. You can look into those books also, because you can hire model, you can repurpose models of ecology in cell biology or some other cases. Hope it clears your questions. Is there any other question from anyone? Okay, you have another question?
Sir, it's my last question.
Come on.
Fallopian tube is
Yeah.
OM travels in fallopian tube. So as we applied the peristaltic wave equation of PDE in our tubular path from mouth to digestive system. So we apply in all tubular paths in our body the peristaltic wave equation there. Can we apply that equation?
I have no idea. I have no idea because I never studied reproductive system of human being beyond class 10 level. Right?
Okay,
so
sure.
It will be audacity for me to comment on. If what you are asking a mechanistic aspect of a biological problem, right? And it will be very wrong for me to mislead you. You should ask somebody who is from physiology background. Even a student who has studied physiology in undergraduate level will be able to help you more than me on this question.
I'll work by myself, sir. Sure.
Yeah, so I am the wrong person to ask this question. I am as ignorant as you in that time. So, anybody else? Okay, so last few minutes, let me say a few things about final examination. In final examination, you will have MCQ type question and you will have short answer type. Short answer type question means you have to do some calculation and then write the value. Many times we write you to write the value to a particular decimal, like for example, third decimal or something. Please follow that instruction. What will be the difficulty level of these questions? The difficulty level of this question will be as difficult as we are putting in the quizzes. Don't expect that questions in the final exam will be very difficult with respect to quizzes. No, it is not like that. Yes, maybe one or two questions may be bit difficult, so that we put those type of one or two questions may be difficult questions, so that the advanced learner can score better than the people who are not advanced, right? So that's fair enough. So we have to put a few questions bit difficult, so that people who are good in the subject can perform better. But otherwise, if you are practicing the additional questions given to you and you are regularly submitting the quizzes, then I can assure you, you will perform well in the final exam also. You must have given the examination for other NPTEL course. It will be the same, computerized one, fully computerized one. And as I said earlier, in this computerized exam, we will not provide you any software for simulation or dynamical system like JSIM, but there can be MCQ question or short answer type question based upon JSIM. So that means you should know how to write JSIM code, how to correct JSIM code. These things you have to know. One thing I will suggest you, even if you have not performed well in some of the quizzes, please look back into the solution that we are providing. Try to solve it. Then I can assure, as I said, if you go through these quizzes, look into the quiz solution, look into the additional questions given, and follow the lectures. We have given you the slide. You will easily sail through the final exam. You do not need to go and study some additional book or something for this course. This course is self-sufficient. The videos, the lecture slide, and the model questions given, and the quiz questions and their answers are good enough for you to prepare for the final exam. So is there any question about the final examination? I'll address them now. We are closing almost to the end, reaching the end of this session. So if you have any question on final examination, please raise your hand and ask them. Yes, Ritika, please go ahead. Unmute yourself.
Good evening, sir.
Good evening.
Uh, Sir, I have filled the form, but I didn't get the application number. So I don't know how to fill those form and how to appear for the exam. This is my first time. I'm very
Okay.
confused regarding this.
Okay. The right person to solve this problem is to write to NPTEL office. Have you written to them?
No, sir.
That is, you have to do not just for my course. Remember, we as an instructor only teach. Everything done on the back end, starting from providing the portal, organizing examinations, and everything is done by the NPTEL office, right? So you should, for all these things, you should always contact them, not for my course only, for any other course. If you ask me again, I will again refer to you to those people only. So don't delay. If you have confusion, today itself drop an email to them, and believe me, although they get hundreds of mails, they are usually very good in replying. You have to be a bit patient with them because they get lots of query, lots of problem happens with people, like multiple things happens, right? And so they get lots of queries, but they do it. They honestly replies and direct the students to the right direction, right? So please write to them today itself. Fine,
Okay,
Ritika?
sir. Okay, thank you.
Don't
Yes, sir.
delay.
Sir, one
Don't delay.
question.
Yeah, one question.
Uh, Sir, I'm from biology background, from botany. My maths is not well. Even I don't know even M of math. Sir, it is very difficult for me to solve those calculus and all. I don't know how will I apply those things in research, in my own research.
Okay. Okay. Yeah, Ritika, I can understand your problem. You are not the only one who has this problem, right? Uh, my suggestion here is not just limited to this course. Please understand that the direction biology is evolving. I must be aware. You must be aware that the way it is becoming data intensive and model intensive. You cannot help, but you have to use mathematics. We have crossed the time when we used to do only ANOVA or t-test. Now you have so many other methods are coming for data analysis because we have large scale data, right? And people are incorporating mathematical model. You pick up any good journal, you will have mathematical model in that. One can argue that, okay, many of the cases, most of the cases possibly, some physicist or engineer or mathematician is building those models as a collaborator and help. As a biologist, do I need to understand them? The answer is yes. See, if you do not understand how a t-test is done, what is the principle behind t-test, what is the difference between t-test and ANOVA, then there is a high chance you will be using a wrong method of statistical analysis while analyzing your experimental data, right? So that's why biologists are also taught to some extent biostatistics so that broadly you understand. So if you do not have good background in mathematics, what I will suggest you, please slowly build up. I am not expecting that you will become a mathematician and build a completely one mathematical model out of the box yourself. But what is important is that maybe throughout your life you will remain an experimental biologist, but you should be able to understand what a mathematical biologist is telling you, what he or she is trying to communicate, what that model means. That you have to understand. And for that, some amount of mathematics is required. In the reading material, extra reading material that I have given in many cases in the below the lectures, you will find some books like Calculus for Biologists. You search in the web, you will find that book, right? So there are some books like these which introduce people of biology background to the basic mathematics, which are essential to survive in this field because the way biology is evolving, right? And also in NPTEL, I believe there are courses where students are biology background are introduced to. These are dedicated courses. I believe I forgot the name. You can look into it, where the basic mathematics are taught to the students. So I will suggest you to go through them, get habituated with it. Remember, you are a biologist, you understand the biology. You should get the skill to communicate with people like me. That's the thing you should target and try. Is that fine, Kritika?
Yes, sir. Thank you so
And
much,
it is,
sir.
and it is doable. Please believe me, you are doable. If you try, you can do it.
Yes, sir. After attending the course, I am confident that I can do.
Yeah,
Thank you so much, sir.
yeah, nice meeting you. So, is there any more question? We have 1 or 2 minutes left. So if anybody has any question, please ask. Okay, as I see there is no more questions. So thank you all for joining me in this live session. Please remember today at 7 o'clock, Akshita will have one live session for problem solving. You must have got the link in the announcement all and also in the email. Please join it. This live session, he will discuss the questions of additional questions of this third week that will help you to prepare for the exam as well as for the quiz. So have a nice evening. See you later in another live session. Thank you for joining with me today. Happy learning.
Thank you, sir.